Again, good morning, grade 10.
Good morning, sir.
Morning, Teacher MJ.
Okay, we're
Morning.
done with the
Morning.
introductory activity. So we need to move forward with the lesson proper, which is the DNA and evolution. Okay, let me share my screen. Can you see my shared screen now?
Yes, sir.
Yes.
Okay, so let's start with DNA and evolution. We will now dig deeper on your knowledge about genetics last grade period or last grade level, which is grade 9, and connect it to evolution. Okay, there is a connection between DNA and evolution. Right? Let's tackle more about this. DNA and protein synthesis. So the DNA or the deoxyribonucleic acid is one of the four macromolecule essential for life. Okay, the DNA is made up of building blocks called nucleotides, which consists of three parts: the deoxyribose, sugar, a phosphate group, and a nitrogenous base pairs or nitrogenous base or bases. Okay, the nitrogenous bases are grouped into two. Okay, the two-ringed purines or purines, adenine and guanine, and the single-ringed pyrimidines or pyrimidines, cytosine and thymine. Okay, let's tackle more about them later on. Can you see my shared screen? Grade
Yeah.
10, can you hear me clearly?
Yes.
Okay. The arrangement of these nucleotides provide the genetic information carried by the chromosomes. Okay, these they are actually responsible for the genetic sequence and the correct sequence of proteins. Remember that these proteins has also blocks. So let's tackle more about the connections of these. DNA contains the sequence that provides all information necessary for proper functioning of an organism. But the actual work of translating the information into a material that is used directly by the cell is done by the ribonucleic acid or the RNA. Okay? RNA serves as the role of being the mediator of DNA and protein. Okay, specifically, RNA dictates what proteins are to be synthesized, becomes part of ribosomes that creates proteins, and moves the amino acid, the building blocks of proteins, to the ribosomes. By the way, what is the protein factor of the cell? Right then, what is the protein factor of the
Ribosome.
cell? Bilosop? Yes?
Ribosome.
Ribosomes. Yes, correct. So that is why, if you're going to look at it, they are actually channeling it to the ribosomes. It could be attached or free ribosomes. Because ribosomes there are considered as the protein factory. So the creation of your proteins are in the ribosomes. These mechanisms involved in the production of proteins from the DNA are parts of the process of protein synthesis. The synthesis of protein is one of the most important events in cells because not only does it provide proteins necessary for structural components of the cell, it also creates enzymes needed to produce the organic macromolecules. Correct. Please read Veloso.
In 1953, James Watson and Francis Crick unveiled the most accurate description of a three-dimensional double helix structure of the DNA molecule. They published their model in a two-page paper that eventually became the cornerstone of modern molecular genetics. The features of their DNA model include
Okay. In 1953, it was because of James Watson and Francis Crick. They actually created the model. They actually observe that the DNA is composed of a double helix. Okay, they publish a paper in two pages, a two-page paper, which actually gives us a feature that DNA is a double helix or double-stranded helix with two strands connected by hydrogen bond. Adenine bases are always paired with thymine. Please take note. I think you're done with this in your grade 9. The complementary bases, grade 10.
We haven't
Ah, sorry. I'm sorry.
tackled it.
So, sige.
It's fine, sir.
Alright. The complementary bases are, when you say complementary, they're usually paired or they actually paired. No other pairing aside from them. Okay? Adenine bases is always paired with thymine. A is to T. And cytosine are always paired with guanine. C is to G or G is to C. T is to A or A is to T. Okay? Which is consistent with the Chargaff's rule that states that the total number of purines in DNA or purines in DNA molecule is equal to the total number of pyrimidines or pyrimidines. Okay? Remember that your adenine and cytosines are purines. Remember, your adenine and guanine are the purines. Cytosine and your thymines are the pyrimidids or pyrimidines. Again, adenine is always paired with thymine and cytosine is always paired with guanine. Okay? A is to T, T is to A, or C is to G, G is to C. The second part of the feature which Crick and Watson actually proposed is the DNA helix is an anti-parallel or anti-parallel, which means that the 5' end of one strand is paired with the 3' end of the complementary strand, or vice versa. If you're going to look at it, they are actually not parallel or anti-parallel, making it a double helix. Okay? You try to look at the illustration. The 5' end is connected to the other 3' end. Okay? And here you can see the pairing, the base pairs. We have thymine and adenine is already paired with a double strand. Guanine and cytosine with a triple strand. Cytosine and guanine, triple strand. Adenine and thymine, double strand. Not only are the DNA base pairs connected by hydrogen bonds, that's the third feature, but also the outer edges of the nitrogen containing bases, which are exposed and are available for hydrogen bonding. Let's move now to, do you have any questions so far with Watson's and
And
Crick?
so far for me.
All right. How about others with Watson's and Crick's um, DNA model? Grade 10? Any other concerns?
Sure, what does it mean by the 5' end and the 3' end?
Okay, let's go back. It is actually the endpoint of the strand. So if you're going to look at the DNA strand, diba, you can actually separate it, right? Are you still with me?
Yes.
Okay. In
Yes.
order to fill the the the, the spaces in between, they actually be they are actually paired through the prime end end part sa kwan ba, sa strand is actually paired with the the three ends of the second part. You know what I mean? Di ba strand, the prime end of the other strand is actually paired in the third prime end of the other strand. So wa siya nagtumong in short. So it can be twisted. That is why it is actually twisted. Because they are anti-parallel. They are not actually parallel. Did I answer your question?
Yes, yes, yes. Thank you.
Any other concern? Any other questions? Any other question?
None for me.
All right. Let's move forward to the RNA. The ribonucleic acid, okay, or RNA, is named after the 5 carbon sugar group that's make up the backbone ribose. Remember that your DNA has a backbone ribonucleic or ribose sugar. Okay, just like your DNA RNA, a sugar phosphate backbone with a nitrogenous base pairs or nitrogen base attached it to make up a nucleotide. Okay, however, RNA is a single is a single stranded and does not have thymine. Instead, it has adenine pairs with nitrogen base uracil. So again, in RNA, instead of having thymine, thymine is not there. Okay, it was actually replaced by uracil. Sure. Why do we need to change thymine into uracil? Okay, remember that your RNA decodes the information. Okay, because it decodes the information, thymine should be replaced by uracil. Anyway, when you go back again, when you have the transfiguration, you will actually convert again uracil into thymines. It's the same thing. But then again, when you're talking about RNA, they actually converted thymine into uracil. RNA may also be found in the nucleus and cytoplasm of the cell. There are three kinds of RNA. Okay, one is the messenger RNA or mRNA. The second one is the ribosomal RNA or the rRNA. The third one is the transfer RNA or tRNA. Okay, they are named after their functions. Okay, all three together with the DNA work to build proteins needed by the body. Remember what I am actually telling you a while ago that RNA decodes the information. Decodes the information for what? For the protein, for a particular protein. That is why. These are the three types of RNA. Okay, the messenger RNA, the characteristic, they're formed actually in the nucleoplasm, that's inside the nucleus, the dark area in the nucleus, with the DNA as template and as groups of three bases called codons, still the same with the DNA. Function brings the code for synthesis of proteins from DNA in the nucleus to the ribosomes in the cytoplasm. And because it is a messenger RNA, it brings now the code for the synthesis of proteins. Okay, what kind of protein to be used or to be created? Just give me a minute. Okay, let's continue. Are you still with me?
Yes, sir.
Okay.
Yes.
The next one is the transfer RNA. Remember that the one that I am telling you a while ago, that they are named after their functions. So it's really easy for you to identify. It's a clover shaped structure with three exposed base called anticodons. So these are now the anticodons. On the one end of the amino acid, on the other found in the cytoplasm. Okay, because this is transfer RNA, they have the anticodons. Okay, it picks up amino acids in the cytoplasm that fits them into proper place in the growing chain of polypeptide inside the ribosome. Okay, this is now inside the ribosomes. It picks up amino acid in the cytoplasm. Remember that the messenger RNA is scattered in the cytoplasm. Remember? Now, the transfer RNA actually connects with the messenger RNA to bring it over to the ribosomes. Okay? The polypeptide inside the ribosomes. Okay? Again, messenger RNA brings the code for the synthesis of proteins. Okay? In the DNA, in the nucleus to the ribosome. Okay, how? This is now the question how. By the transfer RNA. Because the transfer RNA now connects to the messenger RNA. It actually makes anticodon. Anticodon or the other pair of the RNA. Okay? It picks up now amino acids in the cytoplasm that fits them into the proper place in the growing chain of polypeptide inside the ribosome. How about the ribosomal RNA? It is actually made in the nucleolus. That's the dark side or dark area of the RNA. It forms important structural component of the ribosomes. Okay? Again, you will not find it hard simply because it is actually named after their functions. Do you have any questions so far?
None so far.
Okay. Again, messenger RNA form in the nucleoplasm. Okay. The ribosomal RNA formed in the nucleolus, the dark area in the nucleus. Are you familiar with nucleoplasm?
It sounds familiar.
Huh?
It sounds familiar, sure,
Sounds
but
Heard
familiar?
it,
I
but
don't.
don't know the meaning.
Yeah, it sounds familiar.
Okay. It is actually the substance in your nucleus. So if you're going to look at your nucleus, it has nucleolus inside and nucleoplasm, just like your cytoplasm. Cytoplasm is an area in your cell. Okay, that's the cytoplasm. Okay, nucleoplasm. This is now the area of your nucleus. Okay, there are a lot of suspended particles there, like your DNA and your nucleolus and all of those. So it is actually formed there. Okay, once they actually form, or the nucleoplasm actually form these structures, it will be released in the cytoplasm in order for the transfer RNA to create anticodons for that, picks it up, and afterwards transfer it in the ribosomes. Okay. Any other concerns or questions?
None so far for me.
All
None
right.
so far for me.
Hopefully this is clear. Sometimes students usually overthinks about this. This is just very simple. RNA is just to decode information and transfer it to the ribosomes. As simple as that. Okay. Sure. What are those decoded informations? Those decoded informations contained now the sequence for the pro or for a particular protein to be produced. Remember that your ribosomes again, di ba? I asked you a question about um, the protein factor of the cell. That's your ribosome, right? Now, because of this sequence, we are unable to identify a particular protein with that particular set of codons in the RNA. Okay, let's move forward. DNA and its relation to heredity and diversity. Diverse. Okay? When you hear the word heredity, what comes into your mind?
Parents, then
Inherit.
yeah, transfer parents.
Family,
Like they, like the
ancestry.
traits of the parents will be
Wait,
transferred to you.
parents, yes, that's all correct. The transfer of traits, yes, that's all correct. How about diversity?
I think being unique. I'm not so
Diverse,
sure.
correct. What else? What else? Okay. Humans grow and develop according to the instructions in our genes. We are actually developed according to the structures, according to the genes. Remember that your genes are set of instructions. Sure, they're not actually um, the one responsible for making of, like for example, your arms, your limbs. They are not actually the one responsible for it. They're just instruction on how. Like for example, you're building a furniture, for example, a table, a chair. When you build a table or chair, what should be done first?
Design.
Design, yes, correct. Okay, design, the layout. Okay, the design and the layout there are your DNA. Okay, who usually do the tables and chairs after the design? The carpenters, okay,
Builders.
builders, yes. So they are actually the one responsible in making those, just like your ribosomes. Okay, the ribosomes are responsible in making those proteins. So the same concept. Okay, a gene is a segment of DNA that may code for protein of your hair, protein of your like insulin. Insulin is for sugar, which controls the level of glucose or sugar in the blood. The DNA molecule is very long and may contain hundreds or even thousands of genes. The DNA in your genes is responsible for your uniqueness and why you share some features with your family members. This is actually responsible for it. Re- and remember that everyone is unique. Everybody is unique. Okay? When you say unique, even um, even twins are actually different. Have you observed that one? Do you have friends which are twins?
Yes.
Nakabantay mo? They're actually unique. One in another. Like for example, if you can observe twins, other is healthy and other is not so healthy. I'm not telling you that they're not healthy, but not so healthy. Other is thick and other thin. Other is intelligent, the other one is not so intelligent. So because they actually share the same genes, but they are unique. How could that be possible?